Hi everyone, welcome back to the channel. Today we're going to discuss how substances are absorbed into the cell through the plasma membrane. Now, the plasma membrane, more often known as the phospholipid bilayer, serves as a boundary between the intracellular and the extracellular environments of the cell. And now it is selectively permeable, meaning that it allows some substances to pass through while blocking other substances. And the selective permeability of the cell is essential for maintaining the overall homeostasis of the cell, and substances that enter the cell via different mechanisms, such as simple diffusion or cell, really depend on the sort of properties that each molecule has. Now, let's walk through the main methods of absorption into the cell membrane. First method of absorption is simple diffusion. Now, this is a very passive process, meaning that it does not require any energy, and it is a passive process wherein small, nonpolar molecules, more namely oxygen, for instance, or CO2, or other lipophilic molecules, move from an area of high concentration to an area of low concentration down the concentration gradient across the plasma membrane. Now, this movement does not require any energy, and it occurs directly through the phospholipid bilayer. Now, simple diffusion is driven by a random molecular motion process. It does not involve the use of any membrane transport proteins, and it is most effective for small, uncharged molecules that are able to permeate through the hydrophobic core of the membrane without any assistance of specific channels or even carriers. And I think this is a really good time to kind of go over the structure of the phospholipid bilayer. Now, it's composed of a head that is hydrophilic. Hydrophilic meaning hydro, water, philic meaning liking. So hydrophilic head, and it's also composed of a hydrophobic core, hydrophobic tail. So essentially, the head part, the surface part that's in touch with the extracellular or the intracellular environment, is essentially hydrophilic, while the core, the tail composed of the tails, is hydrophobic. Next, we're going to look into facilitated diffusion. And this is another passive process, meaning that it does not require any energy. However, unlike simple diffusion, facilitated diffusion oftentimes requires transport proteins that are embedded into the plasma membrane to assist the molecules that cannot directly diffuse across the lipid bilayer. So it differs from simple diffusion in the sense that it uses channel proteins or carrier proteins that are embedded into the phospholipid bilayer, the cell membrane. Now, these molecules typically include larger polar or charged molecules like glucose, or even ions like potassium, sodium, and chloride, and the channel proteins essentially help these molecules move down their concentration gradient across the membrane. Now, there are mainly two types of proteins that are really involved in this process. First is channel proteins. Second is carrier proteins. Now, channel proteins, which form the pores that allow specific ions or small molecules to pass through, are oftentimes very extensively used in facilitated diffusion. And the second type of protein is carrier protein, and these kind of really bind to the molecules, and they undergo conformational change, and then they release the molecule on the other side of the membrane, the area with low concentration of that specific molecule. Now, an example of facilitated diffusion would be the glucose transporter, which helps glucose move down the cell into the cell when the concentration gradient is higher outside the cell. Okay, now another specific form of facilitated diffusion is the movement of water molecules through aquaporins. So aquaporins, it's essentially a part of facilitated diffusion. And aquaporins are essentially specialized channel proteins that facilitate the movement of water across the plasma membrane. Now, this process is crucial for maintaining the cellular osmotic balance, and this is kind of different from osmosis, and be aware of that. And water molecules, essentially in this process, despite being small, they cannot diffuse freely across the lipid bilayer because of the bilayer's hydrophobic nature, as we investigated earlier on. Thus, we need aquaporins in order to create these hydrophilic channels that essentially allow the water to move down their concentration gradient from an area of lower solute concentration concentration to higher soil concentration or down the water potential gradient. And the selective transport of water is essential for processes such as, for instance, kidney filtration or even the regulation of cell volume. So let's kind of draw that right here. Aquaporins. And this is also a type of channel protein. Carrier proteins. Right, so channel proteins are kind of unidirectional, whilst these carrier proteins are kind of bidirectional. And that brings us to the end of our video. In our next video, we're going to be talking about active transport, endo and exocytosis, the other methods of cell absorption across the plasma membrane.